Okay, so hello and welcome to the second webinar in our series, an introduction to genomics resources at the EBI. So this webinar is targeted at biologists. I hope you're all biologists out there who want to learn more about the resources that EBI provides. I think some knowledge of genomics is a real advantage for this course because I will use some terminology that will be easier to understand if you have this background. And so the major objectives of this webinar then are to provide a high-level overview of the genomics resources that we have on offer here, and also to direct you to more detailed tutorials so you can learn more. Okay, so my talk will cover the following major genomics resources. That is the European Nucleotide Archive, Ensembl and Ensembl Genomes, and the Metagenomics portal. So the European Nucleotide Archive is a database that contains all of the nucleotide sequence data that you go away, do your experiments and submit to us. It's a primary database, which means that it simply contains the raw sequence data that is submitted. Oh, is there a question? No. Okay. That was the ENA. All the databases which are shown below with the icons there, Ensembl, Ensembl Genomes, and Metagenomics portal, are secondary databases, which means they contain data we've added value to and analyzed or integrated with other data. So Ensembl and Ensembl Genomes contain annotation, variation, comparative, regulatory information, and the Metagenomics portal contains analyzed metagenomic sequence data. So why might you want to use our genomics resources? I thought the best way to run this webinar was to think about the main things that you might want to use our resources for. It's not to say that there aren't lots more. I wouldn't be able to fit that in this short webinar, but these are some of the main things which I thought were the kinds of things that you'd want to be doing. So you might want to submit your nucleotide data for publication. You might want to find out where about a specific gene is that you've read about in a paper, find out where it is in its genomic context on the chromosome. You might have generated your own next generation sequence data, and you might want to visualize that in its genomic context and combine it with lots of other data that we know exists. You might also want to get a data set together to work on, either to combine with your own data or maybe just you're doing a completely computational project, maybe a comparative study that needs that. Or maybe you've got metagenomics sequence data and you want to analyze it. If these are the kinds of things that you're after, well, that's great because that's what I'm going to focus on in this talk. We also provide services in lots of other areas too, so do check our website and find out more. I can just see a question, is ENA like Genebank? Yes, that's a very good question. It is indeed. So Genebank's the American version of ENA, and of course ENA is the European version. So a lot of the, we share the data continually with NCBI, so there are duplicate copies, and also the DDBGA in Japan. Okay, so moving on then, the first part of the kinds of scenarios that I gave to you earlier is submitting sequences for publication. I don't know if you're aware of this, maybe a lot of you are already, but in order to publish a study, you really do need to make sure that you make your data publicly available, and it's a requirement of the vast majority of journals now. So you of course, you collect your data. Maybe you've done a fancy sequencing project, get together lots of short reads, and you can submit your data to us. And that data is actually kept private for up to 2 years. So you can submit your data straight away. Certainly we add value to it, so that will be useful for you in your analysis. But we keep it private before you publish. So you've got two years grace period to get your act together and write up that paper. Okay, so where would you submit your sequence data to? And it really depends on the kind of project that you're working on. So the vast majority of projects you would submit to ENA. So maybe you're working on sequencing a new genome that's never been sequenced before and nothing like it. Or maybe you're doing a resequencing project where you've got a reference genome. Maybe it's a metagenomics study or a metatranscriptomics study. Either, well, all of these cases you would submit to the European Nucleotide Archive directly. However, if you're doing functional genomics, so maybe you're doing some kind of RNA-seq experiment where you're measuring transcript abundance in different experimental conditions, in that case you'd submit to ArrayExpress. In fact, the data would then be passed along to ENA in the background, but ArrayExpress would remain the main port of call that you would go for your functional genomics data, both in terms of submission and then retrieving other data that exists in the public domain. Maybe you have been involved in a clinical trial and you've got identifiable patient data. In this case, that data is very confidential, so you must submit it to the European Genome Phenome Archive, where that confidentiality will be maintained. Similarly, so if you've done a population study and you've got lots of variation data in a variant call file, that needs to go to the European Variation Archive. And if you have structural variation, so in large-scale insertions, deletions, genome rearrangements, that needs to go to the database of Genomic Variants Archive. So let's just take the example of ENA, European Nucleotide Archive. What can you submit there? You can submit raw read data. You need to remove the barcodes, so these are artificial sequences which are added to the reads so that you can identify which is which when you pull them together in a multiplex sequencing reaction. You could submit assembled genomes, either partial, so contigs, or slightly larger scaffolds, or even full chromosomes and genomes. And you can submit annotated nucleotide sequences as well. And there's more information there. So what is metadata and why does it matter? I just put this in because it's really important to make sure you include metadata in your submission. It's part of the ENA submission process. So it's a description of your sample and experimental design, including the answers to questions such as how was it sampled, how was it extracted, how was it stored, and so on. I've listed more examples there. But the point is that if you don't include this information, we have minimum standards, but of course the more metadata you can provide, the better. Without it, the raw sequence data is almost meaningless, but with it, it means that people can combine different data sets in the future to do really powerful analyses where you can query for specific conditions. So what do you need to do to submit to ENA? You need to collect together 3 different files. You need your sequence file format, so a little curiosity, a VCF file is an example of metadata. No, VCF file is a variant call file, which is a file that you obtain at the end of like a large-scale population study where you've identified variants, so those typically SNPs which differ from the reference sequence. Whereas metadata, that's the experimental conditions used, so things like the temperature, the location you sampled your sequence from, and so on. I hope that makes sense. Okay, great. Back to this slide then. So to submit to ENA, I say you need 3 different file formats. One is your sequence data, and there's listed list of accepted formats there. Another thing is, yes, the presentation is available online. The other thing you need is the MD5 checksum file, which sounds a strange name, but you can make it very easily using our webbing interface from your sequence file, and the other thing you need is the metadata. We're now moving on to the next section, which is to think about browsing genomes. So where can we go to browse genome data? Ensembl is one of our resources and a comprehensive vertebrate genomics resource. So it contains genomes from many, many different vertebrates, which have been annotated in detail. We also have combined it and integrated it with regulatory, comparative, variation and variation data, including from some large-scale projects such as the ENCODE project, which has lots of regulatory information, and the 1,000 Genomes project, which contains variation data. Ensembl Genomes is also a sister project which where we provide very similar features for a much broader range of species, so those from protists, plants, fungi, bacteria and so on. How do you start browsing Ensembl genome data? Visit the URL at the top, or as Sarah mentioned in our webinar last month, you can go to the EBI main page and then navigate to Ensembl from there. Then you can search for your favorite gene. You can select a result of interest, and you'll get a visualization that looks something a bit like this. But if this is unfamiliar to you, then it will be useful to know how to interpret this visualization. So you can see there's a blue band along here which shows the sequence config. Everything above the blue band, shown here, is then on the forward strand with respect to the sequencing. Do you share annotations with other genome browsers? Yeah, I believe we do, and I mean, the main other genome browser that's available is UCSC, and so they have some of these different lines across here are called tracks, which are ours, and we have some. I mean, the results are the same. Some things are the same and some things are different. You just have to pay attention to the specifics of it, because you can you'll see if you go onto the website and look into this in more detail, you can customize exactly which of these tracks are shown. So some of them are specific to us, and I'll just move on to telling you about these just now. So so one question would be, why are there so many different tracks, so many different genes which are shown here? And that's because some of these are all different gene models, so potential genes. Some of them have been generated automatically, and these are ones which we've generated. We've got a dedicated gene build team that do that. Others have been annotated manually, and again we've got a manual annotation team which do that. In both cases, they're all the annotation is based on biological evidence, so often things like expression data from RNA-seq and other kinds. So, but there's lots and lots of different data that's pulled together, and I don't really want to go into the details of that now. So when both of these automatic and manual annotation are identical, then we colour them in gold, and these are merged. So these are specific to us and would differ from, say, UCSC and NCBI very slightly. But what you'll find actually, certainly for the majority of genomes, is that we get extremely similar results, and you can add different tracks. So I should also just point out, oh, sorry, didn't mean to do that. That the filled boxes, so all of the boxes indicate exons, and filled boxes are coding, whereas the empty boxes, I guess here, are untranslated regions. Okay, so So that was just one view of Ensembl, but actually it's a vast resource and there's loads of different visualizations are available. So I just pulled out 4 which I thought looked particularly pretty. One is that we compute for you phylogenetic gene trees, so phylogeny is based on individual gene sequences. This is one that's shown here. We also do synteny analysis, so this identifies regions of conserved gene order, and you can see them visualized on a map. We show you where SNPs are and what they are and how they differ from the sequenced strand, and we show you where regulatory features are, so things like promoters, enhancers, but also the epigenomic landscape. But what I really wanted to say, I think I mentioned this briefly, but if you want to customize any of these views, you can do if you click on configure this page, and it's really worth exploring that because you'll see there's actually loads of possible data and you can almost customize these diagrams and make them how you want, which is very useful for actually looking in your own research. So and which leads nicely on to how you then upload your own data into Ensembl and visualize it in its genomic context with all this available data at your fingers. To maybe you can even produce images which are suitable for publication from this. So to do so, you select location, which takes you to the region in detail view. So you click on the available genomic location, you select add your data, fill out the web form which looks a bit like this, and then you'll get a result which looks something like this. This was a BAM file that we uploaded, and you can see that you can see nicely the read depth along the chromosome, and you can zoom in as well. If you wanted to zoom in on this, you would just sort of create a box with your mouse, click and drag, and then you could zoom in. But there's a video that explains more detail about that, which I've put just below there. So moving on then, another thing which we mentioned that you might be interested in doing is getting a data set together for an analysis. So this could be potentially quite a large one. You would want lots of the genome and relevant annotation data. And we offer various different file formats as well. You can output this as a sequence file or as a table, depending on what you're planning on doing. And no programming is required. It's really simple. There are just 4 simple steps. So firstly, you select the data set from all of the databases that we have available. Secondly, you filter by various different criteria to restrict what results you get, otherwise you can end up with a very large data set. You select particular attributes of interest, so this is where you decide whether you are interested in the sequence, specific functional annotations and so on which are available, and then you can export the results. But again, I think if you want to spend more time on this, the best thing to do is watch a video tutorial which I've put at the bottom of this slide. So finally then, we'll just move on to metagenomics. So I'm sure most of you are aware that metagenomics is sampling and sequencing various things in our environment that we're interested in. But you would sequence things in a pool, so you don't know what you're going to get, which species, which genes, or anything like that. And so that's what you can find out. You do your sequencing experiment, then we can help you. Our metagenomics portal can provide your quality control steps with your data, so make sure that you only have sequences with low error probabilities, and then we can do various different analyses for you. We can do taxonomic analysis, where you can find out from your sample what's in it, which operational taxonomic units, and potentially their relative abundance. And then also we can perform functional analysis, which means identifying which genes you have present in your sample and what they do. So we can put them into various different functional categories, which again provide you more insight into your question. So how would you use our Metagenomics portal? This is really simple. First you have to submit your data to ENA, which we've already talked about, and then in doing so we provide tools to help you transfer the data and so on. And we also archive the sequence that you send to us. We then perform all of these quality control and filtering steps for your sequences. We analyze the data for you, and then in about sort of a few weeks or so, once you've submitted it, you'll be able to log back into your account and you'll see some really nice, pretty analyses and visualizations. So you can then go away and interpret those and then publish your study. The same rule applies, so it's a 2-year time period between when you submit this data and then when we have to then make it publicly available. Is there a charge? No, it's completely free. So submit as much as you can and overwork our Metagenomics team. Oh, great. Well, I'm glad you like the suggestion of that. Okay, so that brings me to the end of the main part of the talk. I'm just going to draw your attention to a few other things that I haven't mentioned. So as well as these large data resources on offer, we also have tools for genomics. So I've listed some of these here. I'm not going to talk about them now, but you can find them on our website. Here are some useful links. These are the data resources I've talked about. It also works for honesty. Yes, it does. It works for metatranscriptomics. If you're doing functional genomics with an RNA-seq, though, you need to submit to Array Express for that. Last thing as well to say that if you want to navigate yourself and find out a bit more on our website, go to the main EBI homepage, www.ebi.ac.uk. Click firstly on our Services tab, which will bring you to this page. Secondly, to DNA and RNA, and then that will list all of the resources that I've talked about today, a few more, and also lots of tools which I've not mentioned. So I would like to thank our funders very much, thank my team very much. Here are a list of future webinars which are upcoming if you want to put these dates in your calendar. And I'm very happy to take questions. And we really would appreciate your feedback, so please make sure you complete that questionnaire at the end of this webinar. Oh yes, one last thing I should say, you should follow us on Twitter, where you can find out about future webinars and also our free online training courses when they become available. And I think that's everything. So I'm very happy to take your questions now, if there are any more. Oh, you're very welcome. Any other questions? Are there anything? I'd like to know more about NGS. Oh, what do you want to know? So let me just scroll to the start. NGS, next generation sequencing. So there are various different platforms available. Probably Illumina is the most widely used at the moment because it's the cheapest in terms of the amount of data that you get available. The difference between next generation sequencing and previous sequencing methods like Sanger is that it produces these short reads, which just creates lots of extra work for bioinformaticians because you have to piece together these short reads of about 30 base pairs and figure out, and figure out what order they're supposed to come in. And so I think some of the references that I've made earlier today are related to that. I think we have a next generation sequencing course, a video course on Train Online, so I'd recommend that you go and watch that. I'm sure that we can post that available, make it available to you afterwards. Thank you. You're very welcome. And its interpretation. Yeah, the best thing to do is maybe look at our video course on next generation sequence data. Does the variant effect predictor use multiple different algorithms? Yes, it does. There are two main ones which it uses, and I can't remember off the top of my head. I think it's SIFT and Polyphen, and it incorporates sort of sequence conservation data with structural data, I believe. But there is more detailed information available on the Ensembl web page if you just click on VEP. Thank you, you're very welcome. We have Richard has posted that. Thank you, Richard. Thank you, thank you. Oh, that's lovely. You've been a lovely audience. Thank you very much.